Okay, so all right, so it's 1:55. So thank you everybody for being here. I see we have quite a few people on Zoom and here in person. Andrew, I'm just going to hand it right over to you to introduce Carolyn.
Sure. Thank you. And for those of you who don't know me, my name is Andrew Cox. I'm the Avian Research Lead at Florida Fish and Wildlife Conservation Commission. And I'm Carolyn's supervisor and am on her committee. And as I just had said, we weren't sure if Rebecca was going to make it here in time, so I offered to introduce Carolyn, which really is a pleasure for me. I met Carolyn almost the day I walked in the door to FWC in 2014. And I remember she took me out to go band oyster catchers, and it was really quickly very apparent to me that Carolyn was incredibly bright and energetic, driven, and she was just a pleasure to have as an employee. And as she started talking about going back to graduate school, I thought, well, this makes a lot of sense with someone with her skill set. And so in 2016, she did that. She had gotten her undergraduate degree from Auburn, and now she was ready to get an advanced degree, and she did it here at University of Florida. And I remember talking to her about it because she was really excited. And I said, graduate school is exciting. It's also really hard. I'm not so far removed from it that I didn't remember all the long hours, the imperfect mentoring, the imposter syndrome, all of those things that make graduate school really hard. But one thing I didn't think about at the time, because it didn't really apply to me, is that your personal life can really either make a graduate experience better or even harder. And Carolyn has overcome so many challenges in her life over these last 5 or 6 years that her continued progress and her doggedness on this project was even more impressive to me than it would have otherwise been. And anyone who survives graduate school always impresses me by default. So I'm just so impressed and proud of Carolyn, and I'm excited that you guys are here to join her today as she talks about her work with imperiled salt marsh songbird, the Seaside Sparrow. This is a species that is state listed as threatened by Florida, so that's FWC's interest in it. And I hope that you find her work as interesting as I know we will. So Carolyn, take it away. Congratulations in advance, and I'm really looking forward to your talk today.
Thank you, Andrew, for that lovely introduction. Like Andrew said, today I'm going to be talking about my dissertation work with seaside sparrows. Identifying units that effectively represent biodiversity is a central problem in conservation biology. And as the persistence of an increasing number of species is threatened by anthropogenic forces, it's increasingly important that we identify groups that conserve intraspecific biodiversity and the adaptive potential in species. Distinctiveness is a key factor in defining units that preserve these components of biodiversity, and subspecies are often used by conservation agencies to delineate those units that best represent biodiversity at the intraspecific level. Most modern concepts define subspecies as groups of reproductively compatible populations that are separated geographically and have a set of characteristic and distinctive traits. Although these groups have the potential to interbreed, the concept of restricted gene flow between the groups is implied. Delineation of genetically distinct reproductive units is therefore a crucial first step in identifying effective units for conservation. However, a single method of species or even subspecies identification has yet to be proven as reliable across all taxa, and recent studies have shown that adaptive divergence can still take place in spite of ongoing gene flow. Therefore, the use of multiple disciplines such as genetics, morphology, ecology, or behavior has been recommended as a more comprehensive approach when evaluating taxon status or identifying the distinct population segments required for making listing decisions. As such, a combination of genotypic and phenotypic traits can be useful in evaluating the distinctiveness of groups of organisms and have been employed in the differentiation and classification of individuals to groups, populations, and subspecies. So what exactly is a seaside sparrow, you're probably wondering. Seaside sparrows are small passerines and they're extreme habitat specialists. Except for the mirabilis subspecies, the Cape sable, which lives in the freshwater Everglades, they spend their entire lives in salt marsh. They're monogamous. They breed in small isolated populations where they nest 3 to 10 centimeters above the ground, and that grassland that they nest in floods. They require breeding sites located in higher elevation areas with thick vegetation to avoid flooding during the highest tides. Their diet's mostly invertebrates like insects, small crabs, and marine worms that they pick off the surface of the mud, but it sometimes also includes seeds. The species range extends along the Atlantic coastline from Massachusetts to northern Florida and along the entire Gulf of Mexico coast from southern Florida into northern Mexico. Although the species range is wide and the population is stable overall, seaside sparrows are extremely vulnerable to a variety of threats, including habitat loss or degradation due to sea level rise, coastal development, ditching and dredging for harbor deepening or mosquito control activities, and pollutants and oil and chemical spills. The restricted breeding range and non-migratory nature of the Florida populations puts them at even higher risk of rapid extinction events. As we saw with nigrescens and Pilanota, the dusky seaside sparrow and Smyrna seaside sparrow, when they faded into extinction in the 1980s, within a few decades after habitat alterations made their breeding marshes unsuitable. Taxonomy for this species has a complicated history, with different taxonomies recognizing between 7 to 9 subspecies range wide. The Fish and Wildlife Commission currently recognizes 5 remaining subspecies as breeding in Florida. With the exception of the Louisiana seaside sparrow, Fisheri, these populations are not doing well. Mirabilis, the Cape sable seaside sparrow, is federally listed as endangered. The McGillivray seaside sparrow was recently considered for federal listing status, and both the Scott seaside sparrow peninsulae and the Wakulla seaside sparrow junkicola are currently listed by the state of Florida as threatened. But Florida's Gulf Coast populations aren't well studied. Descriptions of the taxa in this area lack clear diagnostic features, and different authors have suggested different range distributions for the junkicola and peninsulae subspecies. Because of this, there's ongoing disagreement as to whether or not peninsulae and junkicola are separate subspecies. In 2013, FWC recommended that genetic relationships of subspecies be re-examined in Florida to better evaluate listing status. A recent study using mitochondrial DNA and microsatellites found no differences between taxa along Florida's Gulf Coast. However, they only sampled 2 Florida populations, and they didn't include individuals from the Western Panhandle. They recommended sampling additional populations and genome-wide sampling before making any conclusions about taxa in the region. To fill this knowledge gap, we tested the hypothesis that there were 2 distinct populations along Florida's Gulf Coast, junkicola and peninsulae, that can be distinguished genetically or phenotypically. We set out to test this hypothesis with the following project goals. First, we wanted to evaluate current population structure to identify genetically distinct units and assess whether these units corresponded with current taxa and their ranges. Second, because differences in phenotype within a species can indicate local adaptation, which can be important for preserving evolutionary potential, we wanted to examine breeding populations of sparrows across their range in Florida for differences in phenotype. And we wanted to see if these differences aligned with subspecific divisions or with genetic units. Before beginning sampling, we needed to locate populations of breeding sparrows throughout the state. We set out to do this using a combination of information from local managers, historic data, and ground truthing. Current information was lacking on the presence of the species over much of Florida's Gulf Coast marsh because these locations are often difficult to get into. Many of these Gulf Coast marshes are extremely shallow with wind-driven tides and littered with limestone pinnacle rock. So We had some pretty interesting experiences getting out there, getting stuck, and breaking our equipment. Overall, we've explored around 400 miles of Florida coastline on foot using airboats or John boats. It was worth it because we found lots of birds for sampling. 14 Florida sites were ultimately chosen. I chose sites throughout the putative ranges of Peninsulae and Junkicola, and I sampled more intensively than previous studies to include areas of potential admixture. Sampling was especially dense in the Big Bend region due to the conflicting range descriptions for Junkicola and Peninsulae in this area. I wanted to make sure that I could clearly define any point of genetic break. Birds were captured within a 10-kilometer radius of a central point located within suitable habitat that contained breeding sparrows. I also added samples from Northeast Florida, Georgia, and Mississippi to assess the genetic relationships of Peninsulae and Junkicola with other Florida subspecies and those in neighboring states. We were unable to include samples from the endangered mirabilis subspecies. All birds were captured using mist nets and call broadcasts, and blood was collected for DNA. The Maritima subspecies is migratory. They're snowbirds, and many of them come down here and winter in our marshes in Florida. To avoid sampling these migrants, we restricted sampling efforts to May through July, and we banded all the birds we captured to avoid double sampling between site visits. We had an overall goal of 20 birds per site, but managed to collect at least 10 samples from each of our sites, although certain sites appeared to have very few birds in contrast to historic accounts. I decided on a RADseq approach and SNPs to achieve our first goal because whole genome sampling methods like RADseq are more likely to capture recent isolation of populations, which is perfect for a subspecies study. It's also cost-effective, and since SNPs are neutral markers, they're not susceptible to the same biases as mitochondrial DNA and microsatellites. This method also samples throughout the genome and yields many more loci for analyses. DNA was extracted from whole blood samples using either a PureGene or DNeasy extraction kit. This extracted DNA was then assessed for quantity, quality, and concentration using a combination of gel electrophoresis, PCR, and fluorometric quantitation prior to library construction. Only samples containing 150 nanograms or more of DNA were used downstream, and all DNA concentrations were normalized prior to library construction. Library preparation followed slightly modified version of the Peterson protocol. Extracted DNA was digested using 2 restriction enzymes. Custom barcodes were ligated to this digestion product, and the ligation products were amplified using Illumina sequencing primers. The resulting adapter barcode combinations consisted of the restriction site, an Illumina adapter, and a barcode between 8 and 14 bases in lengths that differed by at least 4 bases. Successful libraries were pooled at equal volumes, size selected for fractions between 400 and 600 base pairs using ELF-based size selection, and purified using Ampure beads and bioanalyzer at UF's ICBR core sequencing lab. The purified size selected library was then sequenced on 2 high throughput Illumina NextSeq 500 runs at UF ICBR. We ended up with a total of 348 successful libraries. Raw sequence data was checked for read quality and adapter contamination using FastQC, and low quality adapter regions were then trimmed out using CutAdapt. The iPyrad bioinformatic pipeline was then used to demultiplex, filter, and assemble raw sequence reads into loci de novo, meaning that we did not assemble these loci in reference to an existing complete genome. RADseq often leaves missing data in a number of taxa, which can bias analyses. Some loci end up being sampled in only a small number of individuals, which can lead to large amounts of missing data overall in the data set. And so it's common to try and filter out individuals that yield very little data, as well as to filter loci that end up being sampled in a very few individuals. It's also typical in these studies to explore a variety of filtering strategies until a strategy is achieved that reduces the amount of missingness in the data while still providing enough data to answer the questions being asked. So before downstream analysis, I explored the effects of relaxed or strict filtering parameters on sample and loci retention and the amount of missing data. Ultimately, I selected a filtering strategy that retained individuals with at least 400,000 reads and required that loci be sampled in at least 50% of individuals within a population. I selected this assembly because it minimized the amount of missing data and gave me the best coverage across all populations. This left me with 302 individuals, 3,225 SNPs, and 796 loci for downstream analysis. The following population genetics analyses were conducted in R using the listed packages, and Bayesian population assignment was performed using the program FastStructure. I used FastStructure to assess the number of genetic clusters, or K, represented by the data. I did this without consideration of sampling site in the analysis. First, to get an initial look at the data, I used the simple prior setting and ran the model using values of K that ranged from 1 to 16. For each of these, I ran the model using 12 random starts. As recommended for population structure that's difficult to resolve, I then used the logistic prior setting and ran the model for K values 1 through 6 based on these initial results. For each of these, I ran the model for 20 random starts. For both the simple and logistic model, the optimal K across all replicate runs was determined using the Ivano method. And finally, I used the program Compact to align replicate runs and visualize my results. For population level statistics, including estimates of observed heterozygosity and expected heterozygosity, as well as inbreeding coefficient, the populations didn't differ substantially from each other. There was some variation between my sites, but no population seemed to be highly inbred or limited in genetic variation. As you can see here with this heat map, where bluer cells actually represent higher genetic similarity, pairwise FST comparisons supported the existence of 3 genetic clusters: an Atlantic Coast cluster, and you can see here, and a Gulf Coast cluster that includes all these. This Gulf Coast cluster then further separated into a Panhandle cluster and a Peninsula cluster. Atlantic Coast populations were the most differentiated from all other populations. Using the Ivano method, optimal K results from FastStructure suggested K equals 5 best represented the number of genetic clusters in the data. The K equals 5 model suggested 3 main clusters, 1, 2, 3, with admixture of other genotypes in the Peninsula region. K equals 3 was also supported. This model identified a single Atlantic Coast cluster that you can see here, and 2 clusters along the Gulf Coast, 1 in the Peninsula and 1 in the Panhandle. To cluster using a different approach, we also used a discriminant analysis of principal components, or DAPC. Unlike FastStructure, which clusters using the original SNP matrix, DAPC first transforms the data using a PCA and then groups individuals into clusters. Again, we tested multiple values for K, and the K-means clustering identified K equals 3 as the optimal number of K for our data. The DAPC assigned individuals to clusters that, for the most part, as you can see, occupied the same geographic regions that contain genetics groups that were identified by FastStructure. There. Populations that are geographically closer are expected to exchange more migrants and therefore should tend to share neutral genetic markers. When testing for relationships between geographic distance and genetic distance, I found no significant correlation between geographic and genetic distance when considering all populations together, or when considering each genetic cluster separately. The population structure described in the data doesn't appear to be the result of isolation by distance. This would suggest that other factors are driving population structure, such as geographic barriers. To summarize, analysis using genome-wide SNPs showed support for 3 conservation units along Florida's coastline. The 3 genetic clusters identified corresponded to 3 geographic regions: the Atlantic coast, the Gulf Coast Peninsula, and the Western Panhandle. The data didn't suggest the genetic distinctiveness of Junkicola from Peninsulae, and it found birds in the Western Panhandle were genetically similar to those in Mississippi, which are presumably Fisheri. The genetic breaks that we found occurred along the Gulf Coast east and west of the Apalachicola River and between the Atlantic coast and the Gulf Coast. The ranges of several plant and animal taxa also have genetic breaks or range terminations that occur in much the same pattern and in similar locations as the boundaries between our genetic clusters. This phylogeographic pattern has been referred to as the Apalachicola River Basin discontinuity and the maritime Atlantic versus Gulf Coast continuity. This is thought to be the product of changing sea levels that isolated or fragmented populations of plants and animals over time. So on to our next objective, examining differences in phenotype. As part of an integrative taxonomical approach, morphological traits can be useful in evaluating the distinctiveness of groups of organisms. For example, differences in tarsus length or bill size in birds can be important for foraging, fighting, or singing, and differences in plumage pattern may be important for predator avoidance or mate attraction. Differences in these characteristics may affect the fitness of individuals within populations. Because McDonald's previous work in 1989 suggested that Junkicola and Peninsulae could not be distinguished by plumage, we didn't measure plumage characteristics. However, biometric measurements have also often been used in distinguishing and classifying individuals to distinct groups and to subspecies. So we decided to look at differences in morphometrics across the Florida range. Measurements were assembled from live birds collected at genetic sampling sites in Mississippi, Georgia, and Florida. Later, specimens for the Florida Museum collection were added to create a data set that included all subspecies, including extinct ones. Prior to analysis, each individual was assigned to a putative genetic cluster and putative subspecies. Genetic cluster was based on a combination of the capture location and the previously described population structure. Extinct subspecies were not assigned clusters, and all putative subspecific ranges were based on the taxonomic review that was conducted by McDonald in 1988. A series of ANOVAs were conducted on adults to compare the effect of sex, subspecies, and genetic cluster to see whether differences could be found among sampling localities that might indicate regional differences. I also examined variation among sampling sites between groups. I performed both a PCA and a DFA to examine data using both unsupervised and supervised clustering methods. DFA takes a similar approach to PCA, but seeks a linear function that maximizes variation among groups, while PCA maximizes the variance in the data regardless of a priori groupings. I only present summaries of site-wise comparisons due to small sample sizes for certain populations and to avoid confounding results when comparing live birds versus dried museum skins. I didn't include the data set containing museum specimens in downstream analysis. I only summarize these data here. Overall, male seaside sparrows were larger than females. Although there was overlap, I found significant differences between the sexes in all metrics except for bill length. Because of this, we limited all our other analyses to live adult males to avoid confounding results. From now on, I'm going to be, the results are presented from north to south along each coastline as you move left to right across the screen. I found significant differences among genetic cluster and bill length, with bill length differing between all three clusters. However, no significant differences were found in tarsus or wing cord among clusters. There were also significant differences in mean bill length among subspecies. Post-hoc testing revealed these were between McGillivray and all other subspecies and between Peninsulae and Junkicola. There were no significant differences in tarsus length between groups, but there were near significant differences in wing cord between Fisheri and Peninsulae and between Fisheri and McGillivray. Although Junkicola and Peninsulae were significantly different in bill length, all the body measurements that we measured were highly variable across sites. In addition to significant differences in bill length between sites, I also found significant differences in wing cord and a near significant difference in mean tarsus length between sites. Post-hoc testing indicated differences for both metrics were between certain sites, but not others, and these results didn't follow any clear pattern. And as you can see, in the sites that we had many individuals, like here, there was a lot of variation within the site, and there's also a lot of overlap among sites. When we included the museum specimens, which allowed for the sampling of small numbers of individuals from Mirabilis, nigrescens, which is the dusky seaside sparrow, and Pelinota, the results suggested that these subspecies also aren't clearly distinct based on just these measurements. Instead, the results here suggest that there might be a slight trend north to south along each coastline in certain metrics, regardless of the subspecies. For example, if we compare our Mississippi site right here along the Gulf Coast with our southernmost Gulf Coast site down here, or along our Atlantic coast, we compare our Georgia birds with our southern Florida birds, Mirabilis. There's also, though, a lot of variation that's clearly not attributed to latitude. The PCA showed substantial overlap among genetic clusters and among subspecies. And the use of DFA to assign birds to genetic clusters using these traits was only moderately accurate and assigned individuals to the correct group around 66% of the time. I found even lower classification accuracy when using DFA to assign birds to subspecies. And the model only correctly assigned individuals to subspecies 47% of the time. So to summarize, bill length, wing cord, and tarsus length were variable among adult males within and between sites, within and between subspecies, and within between genetic clusters. But this variation did not align consistently across traits in a meaningful way with either subspecies or with population genomic structure. So we conclude that the recognized subspecies, including Junkicola, don't harbor any clear morphological variation based on these metrics that make them important conservation units independent of genetic clustering. We know that broad evolutionary pressures can lead to the conservation of morphologic traits. Because a limited geographic range was sampled in this study, the individuals most likely experience largely similar environmental conditions. This may have contributed to the broad similarities in traits across groups, and it may make it difficult to quantify local adaptation using these metrics alone. Other traits under a higher degree of selection might be better suited to this purpose. So we examined differences in song to see if we could use them to distinguish taxa or genetic units. Divergence in acoustic signaling traits can be an important driver of evolution in birds, especially when they're involved in mate attraction or territorial defense, as is the case with the primary song and seaside sparrows. These differences in behavioral phenotypes sometimes represent important components of local adaptation and can be important for preserving evolutionary potential. Because of this, birdsong has been used to distinguish distinct population segments in bird species. Seaside sparrow song consists of two main parts or phrases: an introductory phrase, which is made up of a series of clicks, pops, and slurs, which is then followed by a terminal trill or buzz phrase. Previous studies found components of the first phrase are highly variable within and between individuals and also between populations. In contrast, the second phrase, the terminal buzz, is consistent and is present in all populations. A recent range-wide study found differences in some characteristics of the terminal buzz between coastal lineages, between certain subspecies, and between genetic groups. But recordings from Florida's coastline were extremely limited, and the Junkicola subspecies wasn't specifically examined. This previous work suggests that the terminal buzz may be useful as a population marker within the species. Male seaside sparrow primary song was recorded at our genetic sampling sites during 2016 and 2017 using the following equipment. Back at the office, Adobe Audition was used to generate spectrograms for each recording session. Then these spectrograms were manually examined and cropped into segments that contain individual bouts of song. The best song bout was then selected for each individual. I then selected 1 second of background noise directly preceding or directly after the song as a noise footprint and used the sample-based filtering noise reduction tool in Audition to further reduce noise in our recordings prior to downstream analysis. Only songs with a high signal to noise ratio where song could clearly be distinguished in the spectrogram without overlapping birdsong or insect noise were selected. Ultimately, we ended up using 74 individuals. Cropped denoise files were uploaded into the chipper pipeline for segmentation and analysis. ANOVAs were conducted to compare the effects of subspecies and genetic cluster. And to see whether differences in song traits might indicate regional differences, I examined variation among sampling sites for each measurement as well. I performed a PCA to see how differences in song metrics clustered without prior group assignment information, and performed a DFA to see how well song metrics could classify individuals into subspecies or genetic clusters. I limited analysis to the terminal buzz portion of the song. Chipper generated the following song metrics for the terminal buzz: mean duration, mean frequency, minimum frequency, and maximum frequency. ANOVAs found significant differences in minimum frequency and mean duration between the Atlantic and Panhandle clusters, and near significant differences in these metrics between Peninsula and Atlantic clusters. But no significant differences were found in any metric between the Panhandle and Peninsula clusters. There were significant differences in minimum frequency between McGillivray and Peninsulae, and near significant differences in duration between McGillivray and all other subspecies. but not in any metric between Junkicola and Peninsulae. All song metrics were highly variable across sites, and post hoc testing revealed significant differences between some sites, but not between others, again, with no clear pattern. PCA showed substantial overlap among genetic clusters, among subspecies, and among sites. Although most individuals from the two southernmost sites were slightly differentiated from other sites, overall, our individuals occurred along a continuous gradient when they were classified using these traits. The use of DFA to assign individuals to clusters was unreliable and only assigned individuals to cluster correctly 54% of the time. I found even lower accuracy when using DFA to assign birds to subspecies at 43%. So to sum up, I was unable to distinguish between subspecies or genetic clusters in Seaside Sparrow using these song characteristics. And as such, I couldn't find any support for the distinctiveness of Junkicola using the differences in song. It's possible that site-specific factors rather than genetic factors might be driving the differences in song that I found in the Florida range. Our two southernmost sites had the highest minimum and lowest maximum frequency and were audibly distinct from our other sites. I'm going to play a few songs for you so you can hear the difference. Here's a bird from St. Mark's National Wildlife Refuge, which is basically south of Tallahassee, that has a bird with the more typical terminal buzz. And this is a bird from Cedar Key, also with a typical terminal buzz. Lastly, this is a bird from the southernmost site, Werner Boyce Salt Springs, that has the unusual terminal buzz. You can hear that it's really different from the last two. Birds are known to adjust the characteristics of their song to optimize transmission. And although we didn't measure it, I noted that the habitat's very different at these two southern sites with respect to others' sites. These two sites had louder anthropogenic noise levels from traffic, airplanes, and all kinds of stuff like that, airboats. And it was predominantly small patches of marsh surrounded by mangrove, like this, rather than the more open marshes that we found at other sites, like this one. So it's possible that habitat differences are driving the differences in song that we found using our data, but more study would be needed to find out what's really going on. So to sum things up, we found evidence for 3 reproductive units along Florida's coastline using genome-wide SNPs. 1 unit along the Atlantic coast and 2 units along the Gulf Coast, 1 east and 1 west of the Apalachicola River. We found no genetic or phenotypic evidence for the distinctiveness of Junkicola in this study. And I wanted to thank FWC for providing funding and support for this project. And I want to thank the agencies who provided access to lands under their jurisdiction. Thanks to Andrew Cox for being my constant advocate. Also, thank you to my advisor, Rebecca Kimball, and to the rest of my committee, Jim Austin, Abby Powell, and Sabrina Taylor. I thank all the members of the Kimball Braun Lab, especially at Concha Pandy, for helping me with bioinformatics, and thanks to the McDaniel Lab for helping me with library construction. I also want to say a special thank you to Eric Fortman, my crew leader, and the countless volunteers, interns, and FWC staff who brave mud, sun, and bugs just to catch a few birds. And last but not least, I want to thank my friends and my family for their love and support throughout my time at UF. Does anybody have questions?
Hey, okay, yeah, thank you for that. Okay, so I can monitor in the room, and I'll also help you monitor the chat too.
Okay.
Yes, we got a question back there.
Hi, I really enjoyed your talk. I have a quick question about when you were talking about your genomic methods. You mentioned that the birds were monogamous, and I guess what I was wondering about was there any possibility that the snowbirds could have been hybridizing at any point? I don't really know the seasonality of those birds, but I guess that was like one area that popped in my head of potential like gene flow, if there was a chance that any of the snowbirds at any point could have hybridized with any of the Florida groups.
So I know there's no real evidence of it, but I know there's a lot of other species in Florida that do weird things, like they're migrating from another state and then they come down and then some will stay here. So I wouldn't doubt it if it happens rarely, but I do know that for the most part, the species, they're pretty philopatric once they settle down. And we would be able to tell when migrants had left the area when we were trapping, because other migratory species would all kind of disappear at the same time, if that makes sense.
So thank you.
Okay.
And Esteban has his hand raised.
Hi, thanks for your talk. It was great. One question. So currently, well, or probably many questions. Currently, Yunquicola is accepted as a subspecies, right?
Right.
However, the genetic analysis of your work doesn't support that.
Right.
If it had the results been different, at which, and this is probably difficult and perhaps there isn't necessarily a consensus, where would you have said, okay, the genetic analysis, yes, it does support that? At which point, at which amount of difference, right? Would that genetic analysis in itself contribute to the idea that this is a subspecies?
Right now, I know U.S. Fish and Wildlife Service likes, I guess that's a bad word, they like to use genetics over some of the other things. They prefer that. And they're sort of, I think, still in the process of trying to work out other characteristics and like what degree of distinctness or similarity that it would need to be. I don't think anything's really set in stone yet.
Okay. Yeah. And for the moment, the consensus, I don't know when this happened, but at some point, the consensus became that it was a subspecies, and at the moment, it's still taken as such.
Well, so some taxonomies list it as a distinct subspecies, and some don't. It depends, but that's kind of the thing. Like the agency recognizes it as a separate subspecies, and this was the first time anybody had actually really examined the genetics of those populations. So
awesome, great work, thank you.
Yes, we got a question here. If you just press like the edge of it, and then you'll see the green light turn on.
There we go.
Yes.
Hi, Carolyn. Interesting talk. I have a question for you on the plant side. You mentioned mangroves there.
Yes.
And so here you have this threatened, potentially endangered species now potentially being threatened by a protected species. So, where is this headed with the range change of mangroves that is currently occurring, within Florida?
So I'm assuming that as mangroves fill in the marsh, there's going to be fewer birds. There's definitely fewer birds in those sites down south that were mangrovey than in some of the other sites. But yeah, I mean, as climate change happens and sea level rises and all that kind of stuff happens, the birds are kind of stuck between a rock and a hard place, in my mind, where they're just not really going to have anywhere to go in a lot of places. So does that answer your question?
It's a challenging thing we wrestle with, but thank you.
Any other questions for Carolyn? Okay, well, Carolyn, that was wonderful, really interesting. And thank you so much for being here. And Andrew, thank you for introducing Carolyn. That was great as well. And I believe, Carolyn, I think your contact information is in our flyer that goes out every week. So if people do think of questions later, maybe they could email you or get in contact. But thank you very, very much. And so for everybody who is enrolled here or just in general needs to get in touch with me, obviously, you know where to find me. Otherwise, I will see you all on Monday. Thank you.